Hello. Today I'd like to talk to you about blood types, blood transfusions, tissue and organ transplantation. You guys are probably familiar with the concept of a blood transfusion. If somebody has lost a lot of blood, they will require replacement of that blood because your red blood cells perform an important function of carrying oxygen to a tissue. So if those go too low, you're in trouble. Also, your blood volume is important for the function of your cardiovascular system. There are cases where you need blood transfusions. However, if a blood transfusion donor and a recipient are not matched properly, you can get a transfusion reaction. And these can be very negative. They can be anywhere from your red blood cells clumping together and messing up your circulatory system, or your blood cells lysing, and basically you're becoming suddenly super anemic, or even can cause death in some cases. So it's very important that a blood cell recipient and a donor are matched properly. What determines whether or not these blood cell donor and recipients are matched are what types of antigens are present on a person's red blood cells. So there are over 30 total antigens that can be present on your red blood cells. Most of these are pretty minor, have a pretty minor contribution. They are not really something you need to worry about for donor compatibility. There are two main classes of red blood cell antigens that are very important determinant for whether or not a donor and a recipient are compatible. And these are ABO blood antigens and RH blood antigens. So we're going to go into detail about all of the different ways in which we think about these antigens. And the first thing we want to talk about is what determines somebody's ABO blood type. So you have these ABO antigens, and these are encoded by genes. And they're actually all encoded by one gene. Which allele you have at this one genetic locus will determine whether or not you have A blood, B blood, AB blood, or O blood. You have different alleles, as I mentioned. You have an A allele, a B allele, and both of these are dominant alleles. In fact, they can be co-dominant, which means that if you have both, they can both be dominant. So you have AB blood. You have a recessive O allele, which means that you actually need two copies of the O allele in order to have O blood. But if you only have one copy, you're going to have either A or B blood. Let's say you have two people who are going to reproduce. One person has A-type blood and one person has B-type blood. And the type of blood that their offspring will have is going to depend on what their genotype is. So let's say this person who has A-type blood has AO genotype, and the person who has B-type blood has BO. In that case, if we draw out our handy-dandy Punnett square, we can see that there are 4 different possibilities of blood types in their offspring. You could have one child that has AB. You could have a child that has BO. You could have a child that has AO, or that's OO. So you could have AB blood, B blood, A blood, or O-type blood. All are equally possible in this scenario. Your genotype really determines what your phenotype is going to be, and also potentially the phenotype of your offspring. So that's the genetics of this. But what are the kind of molecular determinants of your ABO blood type? What we really mean when we say your A blood type or B blood type are the presence of these specific antigens. So, for example, somebody who has A blood has this circular A antigen. Someone who has B blood has this, diamond-shaped B antigen on their red blood cells. And if you have AB blood, you have both. You have both A and B antigen. Whereas if you have O-type blood, you actually don't have any antigen. So you have no antigen here. This is just kind of the basics of this. But let's go into the actual molecular details of what these antigens mean. And this will also give us an idea of why you have dominance or recessiveness of these different alleles. What are these antigens? They're all actually polysaccharides, which are just little 6-chain sugars stuck together. So every single person, regardless of their ABO blood type, has something called an H group, which is just these 4 sugars stuck together in a chain. Now, on top of that 4-sugar chain, you can have added an additional sugar depending on depending on the type of sugar you have, that's going to determine whether you have A blood or B blood. So we have this last sugar that we're adding, and in the case of B blood, it's called galactose, and it's a 6-chain sugar shown molecularly here. Now, if you have A blood, you actually have a very similar sugar. So you can see almost all of this is the same. The only major difference is it has this kind of extra bump, molecular bump attached to it. And that's called N-acetyl galactosamine. And so B blood has the galactose attached to the H group, whereas A blood has the N-acetyl galactosamine attached to the H group. And AB blood has both the A version and the B version, all in the same within the same red blood cell. And the way that you get this difference, which seems like a kind of a small molecular difference, comes down to the enzyme that puts on these extra sugars. In the case of your B blood type, your enzyme binds really well to galactose, that last sugar, to put it onto your H group. But in the case of your A blood, it has a little bit of a different three-dimensional structure that allows for that extra molecular bump. So you have this little extra molecular bump that can fit into the A-type enzyme. And in the case of the O-type blood enzyme, what you actually have is a very small shortened SAD enzyme. And this shortened SAD enzyme can't actually put any sugar on. This gets to the mechanism by which you can have genetic codominance, because if you have both the B-type enzyme and the A-type enzyme, you're going to simultaneously be putting on either this or this on a given H group within your red blood cells. And it also explains why those are dominant over your O enzyme, because your O enzyme is really just the absence of that enzyme's activity. So if you have one copy of the enzyme, it will already give you that A group or B group being attached. Just to give you an idea of the kind of ways in which we figure these things out in science, the ABO blood type has been described and known about for a very long time. The original paper that described it is in German, and it's from 1900. Even though we've known for a very long time that these blood types exist, we didn't always understand really the molecular basis for why this was happening. And it wasn't actually until 2002 when this structure of this enzyme was published, the enzyme that puts on that last sugar, that we really understand the molecular basis for the difference between A and B blood specificity. This is a figure from that paper, and it's showing you the active site of your enzyme. So this is for your A enzyme. And it has these amino acids, these 3 amino acids at the active site. And these 3 amino acids are important for determining the specificity of your enzyme for a specific substrate. For example, you have this leucine here. And leucine's a pretty small amino acid. And this is distinct from your B blood type active site, enzyme active site, because at that same position, it has a methionine, which is a pretty big amino acid. And this methionine is kind of bumping out, jutting out, and that's going to prevent that extra molecular bump here. So our A-type substrate can't bind to this B-type site because of this extra bump here. But because the A-type enzyme has a leucine here, which allows for this extra bump, it's basically specific for this A antigen substrate, whereas this B-type enzyme is specific for the B-type antigen substrate. So I just want you to appreciate some of the ways in which, these bigger physiological things really are determined by these small molecular changes. In this case, it's really just one amino acid difference in a protein that's determining something that plays a big role in, whether or not you can get a blood transfusion right away at a clinic. Now I want to talk to you a little bit about not just the antigens, but some of the idea behind an immune reaction. Because we said that a transfusion or reaction is basically a recipient's immune system reacting negatively to the transfused red blood cells of a donor. And the reason this happens is because we have to think about what kind of antibodies will our body make against these antigens, given the type of antigens that we actually have present in our blood. So I want to go through the chart again that we talked about the antigens present, but now I want to talk about what antibodies might be present in each person's blood. So, for example, if you have A blood, you have these A antigens on your red blood cells, but you do not have B antigens, right? So when we talked about our B cell selection, we said that B cells that have auto reactivity, that are basically would cause autoimmune reactions, those get killed. But in this case, our antibodies that are against the B antigen, those B cells will not be selected against because we have no B antigen present. So what happens is people who have A blood will make B-type antibodies generally. It's kind of a fun principle of immunity in general that, if you can produce an antibody, your body often will, unless it's negatively selected against because of that, crazy combinatorial complexity that we talked about that can generate a lot of antibody diversity. So people with A blood have B antibodies, and kind of, on the flip side, people with B-type blood, because they have B antigen but no A antigen, they produce anti-A antibodies. And people who have both A and B alleles, they have AB blood, they have both A and B antigens, they're basically not going to make any antibodies against them, because if they made anti-A antibody, they would react with their own blood cells. If they made anti-B antibodies, that would react with their own blood cells. So they don't produce any antibodies. And O group, kind of also the flip side of AB, they don't have any antigens. No A, no B, no nothing. And so their bodies can produce A antibodies and B antibodies with no autoimmune consequence. So they often have both anti-A and anti-B antibodies. And so the fact that these antibodies exist in a recipient's bloodstream, in their plasma, will determine what type of blood type of a donor you can receive blood from without getting an immune reaction. Okay, so let's talk about how you know your blood is A blood or B blood. And it's actually following the principle of immune reaction, because the way that you detect it is by trying to cause an immune reaction. So blood typing procedure means you take some blood and you actually mix it with anti-A or anti-B antibody. And you see, do you see a negative reaction? And the reaction that you're actually looking for is your red blood cells clumping together, because the antibody will bind to multiple, because antibodies have two binding sites, they can bind to multiple red blood cells. So all your red blood cells will kind of stick together, and you'll get this kind of weird, little dots of what they call it is agglutination, which is just a fancy way of saying clumping. So if we have O-type blood, for example, we don't have any antigens present, so our blood's going to have no reaction. It's going to be negative for both the reaction with anti-A and anti-B antibodies. If you have A blood, because you have A antigen, you will have a reaction with anti-A, but no reaction with anti-B. If you have B blood, you have a reaction with anti-B, but no reaction with anti-A. And if you have AB blood, you'll have a reaction with both anti-A and anti-B. So this is how we determine blood type. Now, how do we figure out who's compatible? Well, it's basically the same principles that we've been talking about up until this point. What type of antigens are present on the donor's blood versus what type of antibodies are present inside the recipient. So by that principle, and we're basically going to go over this chart of looking at, what combinations of donors and recipients work well together and which ones are really bad. So you have a recipient, let's say AB, ABO, all the different combinations of blood types. You have your donor, same combination of blood types. And you know that if you have the exact same blood type, which is basically this diagonal here, you're compatible. So if you have A blood, you can donate to A blood. If you have B blood, you can donate to B blood. If you have A-type blood and we're looking at a B-type donor, that's not compatible. Reason being is because people with A-type blood are able to produce B-type antibodies. So if you have B type antibodies in your blood, those are going to react with the red blood cells from your B type donor, and that's going to basically cause a negative transfusion reaction. You don't want that to happen, so we won't do that. It's very similar for AB blood. Because the people who have AB blood also have B type antigen, you don't want blood from them. Now, what's interesting comes up is when you talk about an O donor. So an O donor, because they don't have any antigens on their blood, any A or B antigens on their red blood cells, the O-type donor can donate to an A recipient, because even if the A recipient makes anti-B antibodies, they're not going to react with an O-type donor's red blood cells. This is actually the reason why people with O-type blood are called universal donors, because they have no antigens on their blood. So regardless of whatever antibodies your body produces, if you have A, B, or O blood, it doesn't matter, because those are not going to cause negative reactions with a red blood cell that doesn't have any antigens. So O blood can basically donate to every single blood type. And then the other kind of interesting case comes from AB blood, because people who have AB blood, remember, they have A and B antigens on their red blood cells, which means they can't produce any antibodies against either of those A or B antigen. And because they can't produce any antibodies against A or B antigen, they can accept blood from all the blood types. From A, B, doesn't matter. And that's why they're called the universal recipient, because no matter what happens, they're not going to make antibodies against another red blood cell type, because it would attack their own red blood cells as well. And so there are a few, cases where you can either donate to anyone or accept blood from anybody. But it's important to note that not all of these things are equivalent. So, for example, if you have AB blood and you're getting blood from either an A donor or a B donor, these A donors, they make anti-B antibody, and these B donors make anti-A antibody. So you're going to transfuse blood that might have some level of antibody production against the recipient's own AB blood cells, red blood cells. Now, this isn't generally a problem. This is really considered a minor mismatch. The reason being is that, generally when you're getting a blood transfusion from a donor, you're not getting that much blood from them. They're giving you a small amount of blood compared to the amount of blood that you already have in your body. So you're diluting their blood a lot. And these anti-A and anti-B antibodies generally are not super high concentration in the blood. So it's not generally a problem, but if you have a decision to make, it's better to give an AB recipient blood from either an AB donor or an O donor, because they're not a minor mismatch in the same way that they would for an A or B. So that's talking to you guys about these transfusion compatibility for ABO blood groups. And as we said, this is just kind of a review that, when you have red blood cells that have antigens that can bind to a recipient's antibodies, not only do you get the clumping that we talked about in the blood typing procedure, but you can also get lysis because of what we talked about last week with the immune reactions. When you have an antibody binding to an antigen, you can have either a macrophage come in and chew it up, or you can have the complement cascade come in and put a big hole in the membrane of that cell. So we really want to avoid these negative transfusion reactions. So that was all I wanted to tell you guys about the ABO type antigen system. But now I want to talk to you about the other system that we mentioned, which is the RH antigen system. And RH is another important antigen for blood to consider for blood transfusion. And this is actually only a single type of antigen called the D antigen on your red blood cells. So if you're Rh positive, you have the D antigen present on your red blood cells. And if you're Rh negative, you do not have any D antigen on the surface of your red blood cells. And this is a slightly different genetic scenario because you only have two alleles. You have one dominant, capital R allele that would give you the D antigen. That's dominant. And then you have your kind of lowercase r allele that is recessive. So if you have that, it's unless you have two copies of that, you'll have Rh positive blood. So if you have, for example, a woman who is Rh negative reproducing with a man that is Rh positive, and it depends on what his genetic makeup is. So it could be that he's got one dominant allele and one recessive. If that is the case, then you have a 50-50 shot of having a child with Rh positive. The reason why this is of note is because unlike the ABO system, where you generally a person who has A blood will generally have anti-B antibodies in their blood system. For the D antigen, you don't generally produce D antigen antibodies regularly. The reason why most people would produce a D antigen is if they've been exposed to Rh positive blood. So if you're Rh negative, even though you're Rh negative, you don't generally have antibodies against the D antigen unless you've somehow been introduced to the Rh positive blood. And it's relevant specifically in the case of pregnancy. So that's why we sort of took you through that genetic scenario. So let's say you're an Rh negative mother and you're reproducing with somebody who is Rh positive. Then you have at least a 50-50 shot of having a child who is Rh positive. And because of the nature of the, of pregnancy, the D antigens from your child will pass through the placenta into your bloodstream. So you're going to get exposed to that D-type antigen. And now your body's going to start making those antibodies against the D-type antigen. And when that happens, especially during birth, when your baby gets exposed to a lot of, potentially exposed to a lot of antibodies from mom's blood, baby will receive those antibodies, and those antibodies will basically cause a negative, not a transfusion reaction in this case, but a negative reaction with your newborn's red blood cells. So this can cause a syndrome called hemolytic disease of the newborn, where the baby's red blood cells will basically either lyse or get destroyed by the antibodies that are coming from the mother's immune system. And so this is another thing you want to consider, and this is another reason why you definitely don't want to put Rh positive blood into an Rh negative donor, because that would introduce these D antigens that would cause the creation of these antibodies. The last thing I want to talk to you about is sort of related to blood types and blood transfusion, but it's basically tissue and organ transplantation. And it follows a lot of the same principles that we've already discussed, which is that when you give somebody an organ from a donor, you want to make sure that the donor and the recipient are well matched. And the first step of matching a donor and a recipient is really matching them via their ABO blood type. So you need to make sure that they're ABO compatible first. And if they are ABO compatible, that's good. You can go to the next step of figuring out their compatibility. The second level of compatibility is really figuring out how compatible their tissues are. So this will go into detail a little bit, but it's rare to be a perfect match. If you're even a partial match, you're going to go to the next level, which is to transplant the organ into the recipient. And this is it's not a process that works 100% of the time. It might be successful, it might not be. If it is successful, the next step is really to give the recipient immunosuppressive drugs, and they have to stay on those immunosuppressive drugs for the rest of their lives. As we've talked about in the immune lecture, your immune system is constantly patrolling for foreign antigens. So it's very unlikely that the person, unless they're your identical twin, they'll be an exact match with you. So if there are any presentation of antigens from that donor organ, then your body would actually reject the immune, or it would basically create an immune reaction against that organ, and then you lose that organ. You have to stay on immunosuppressive drugs for indefinitely when you have an organ transplant. So I want to talk to you a little more about this tissue typing procedure and how we figure out, if somebody is a match. This will kind of harken back to our previous lecture where we talked about MHC, because the genes that you actually care about when you're thinking about tissue typing are the MHC class 1 and the MHC class 2 genes. These were the genes that are the proteins that are responsible for presenting antigens to T cells. In our bodies, we have 2 different MHC class 1 genes, and we also have one MHC class 2 gene. And so we have 3 different genes, but each of us inherits a copy of genes from our mom and our dad. So 6 total alleles of these 3 genes. And because these are antigen binding genes, they're highly variable in terms of their sequence. They don't go through VDJ recombination the same way that our T cell receptors do, but they are highly variable locus. So there are over 150 known alleles at these 3 genes. That means that there's a lot of complexity in trying to match a donor to a recipient. You only have a 1 in 100,000 chance of matching, of being a perfect match in these 6 markers. So you can sort of tell from here that most people who get an organ transplant from someone else are really likely to be only a partial match, which kind of reiterates the idea that you really have to be on immunosuppressive drugs for a long time if you have an organ transplant, because it's likely that you're not perfectly matched with them, so you have to prevent the recipient's immune system from negatively reacting to the donor's organ. So that's all I'd like to talk to you about blood types and transfusions and transplantation. And that's really the end of our blood series in general. So I hope you enjoyed learning about blood, and we're going to start covering kidneys and urine formation next.